We are back after 5 days with a new video regarding this fluorescent microscopy, the fluorescent staining. This is our next tutorial video. This is our ninth tutorial video. This is our fluorescent staining. Here in this case, we are using a fungus called Malabar spinach. This is a cercospora fungus. So we are going to stain this cercospora fungus in fluorescent stain. So what we need for this is the fungus sample. As you can see, it is in the culture flask, a slide, common glass slide, a staining box. This is our staining box. You can prepare it by your hand only. This is a just simple plastic box with two aluminum rods fitted to keep the slide on top of it. And now the important stains are the auramine rhodamine stain and the indigo caramine rhodamine phenolic confocal stain. And another one is the bluing agent. This is very important. This is the ammonia solution. Here we are using the bluing agent as ammonia solution. This ammonia solution is nothing. The simple ammonium hydroxide solution, 200 micro ml of ammonium hydroxide solution in 100 ml of water. So this is the concentration of that bluing agent, and therefore we are going to prepare this slide. And for this, another thing we need is the fixation of this slide. This is formal saline, and we will be using diethyl ether for fixation of the slide. So please watch this, start watching this video. We are going to prepare this slide, and we are going to do the first we will do the fixation, and then we will go for the staining, because this fixation is very important. Because without fixation, if you go for fluorescent staining, the staining will be very bad, and the specimen will get ruptured. The cells of the specimen will get ruptured, so we won't be able to visualize the structure properly. So this staining is necessary this fixation is necessary very well. For that, we are using a formal saline solution and diethyl solution. So we are going to use that and fixation process and all that. So let us begin the process. First of all, we have to separate this fungus from this fungus media by centrifuge around 11,000 rpm. Then we are going to fix this solution, that sample, this specimen with this formal saline and diethyl ether, and then you will begin to centrifuge the process, and that process will be done on the 2,500 rpm again. And then we will remove the mass, and we will get the fungus media. We will take that fungus media on the slide. 20 micromole of that fungus media will be taken on the slide. And after that, we will be staining this rhodamine stain and the counter stain, the confocal stain, the ISP stain, and then we will do it, and we will dry and see under fluorescence microscope. So let us begin the whole experiment. And so this is our process. So let us begin the process, and you see that how the process is going. So friend, we will get our one centrifuge tube, and this is a 2 ml centrifuge tube. As you can see, it would be clean and dry tube. Let us take a clean and dry tube, and this is our clean and dry tube, and we will take around 2 ml or rather 1.8 ml or 2 ml of this base of this fungus media by the autopipette, and that we will transfer into the 2 ml tube. This is a very thick mass, as you can see. So this we have to take it, and already it is there. So we will just remove the air bubbles, and we will centrifuge it around 11,000 rpm. So after that, we will be fixing it, go for fixation. So let us remove this fungus matter over here, and let us put it in the centrifuge and see how it goes. So this is a centrifuge tube, as you can see in the camera, the second camera, and this is, we are going to centrifuge under 11,000 rpm. So friends, we are ready with the fungal separation. As you can see in the centrifuge tube, our fungus is separated. Here you will be able to see the tube. That is how fungus has been separated, and at the bottom there is the fungal base and the culture media is at the top. So this we are going to separate the culture media. So here we are going to take a small pipette. This is our small pipette, small 1 ml pipette. This is a small 1 ml pipette, and 1 micro ml, 1000 micro ml pipette, and we will slowly separate the culture media. This you can discard. This is not needed to keep. So as much as possible you can discard. So it is done, and now we are going to use this fungal base, this formal saline base. So here is our formal saline, and this is our formal saline, and we are going to take around 500 micro ml of this formal saline. This is around 500 micro ml of this formal saline. Please keep in mind these separations. Please keep in mind these separations. So these separations you should keep in mind. These separations you should keep in mind. Otherwise, these formations will be bad. So what we are going to add is that we are going to add 500 ml of this, 500 micro ml of this formal saline. This is 500 micro ml, not 500 ml. This is 500 micro ml of this saline. So this is the saline, formal saline. And now we are going to add another 500 ml of this diethyl ether. So bottle has become tight. Okay, now. So we are going to add around 500 ml of diethyl ether. Okay. Now we have to shake the test tube, shake the centrifuge tube vigorously. Just discard the tip and with the pipette, we have to vigorously it does not need to centrifuge as it is a very dense fungus, so we need not to separate the solution. So just we will discard the solution and this fungal base. Okay, now it is discarded. So what we are going to do is that now our fungal base is fixed. So you can see it is fixed. In this camera you can see it is fixed. So let the camera focus and you will see the fungal base. So this fungal base. So now we are going to take around 20 micro ml of this fungal base. Now we are going to use another photopipette. This is our 20 micro ml photopipette, and a very small tip that we have shown you previously in our photopipette videos. So this is our separated fungal base. And little of that we will take. This is our fungal base, and we are going to take. So this is our fungal base over there. So rest we will discard. And this we are going to use a slide. Another slide, take another slide, and just hold the slide like this. Hold the slide like this in the front of the camera. You just hold the slide like this. So it is not coming in the camera. So this is the camera. Hold the slide and just move upwards and downwards, then again upwards and downwards, and upwards and downwards, like that. Only 2-3 moves, not much. So this is our slide is done. So this now we will keep for dryness in our dry cabinet. This is our dry cabinet. This is our dry cabinet. We will just put it like to the dryness and let it dry for 1 or 2 minutes. So we are ready now with the slide. You can see the slide is completely dried in this camera. You can see the slide is completely dried. So now we are going to stain. So please watch this video, this process very carefully. Now this is our auramine or rhodamine stain. This stain has been prepared by making a 1 gram of auramine red powder and rhodamine fluorescent powder, auramine stain powder, which is available. And this is another one. Second one is rhodamine stain. These two powders these two dust solids are available. These two are in a powder form. These are available around 25 grams package. They are available, and both are mixed in 50-50 ratio. So in 100 ml of water, 100 ml of distilled water, and keep it for ready stain. Now this is your fluorescent stain. This is our main fluorescent stain. And the next one is indigo caramine, rhodamine, phenolic stain. So these are all. Indigo caramine is one indicator, the rhodamine is another indicator, and phenol. This is the third element for coagulation. This is the coagulating element. This is the phenol, and the rest part is water. So these are mixed in 1 gram ratio. 1 gram of indigo caramine and rhodamine. You can mix 500 milligram also. You can mix 500 milligram of indigo caramine, 500 milligram of rhodamine, and around 1 gram or 2 grams of phenol in distilled water, 100 ml of distilled water, and your stain will be ready. So, and the ammonia, as I have told, this is the bluing stain. This is 200 micro ml of ammonia in 100 ml of water, distilled water. So keep in mind, this should be perfect distilled water. But if the distilled water is wrong, your whole thing will be wrong. So be careful about this distilled water. So these three things will be ready for your microscope, for your fluorescent staining. Now watch for staining. Now, with the help of a 1 ml pipette, we are going to start the staining process. The staining process is very easy. Just we have to take around 1 ml of just cover the stain. This is to cover the stain and cover this specimen with the stain. So this is around 1 ml. It will take around 1 ml or 1.5 ml. So this is our stain is done. Now we have to wait for 2 minutes or 3 minutes to get this stain fixed. This stain here to put the specimen to take up the stain. So this is our first stain. So what we did the auramine rhodamine stain mixture to this slide in the distilled water, the solution, the stain solution. We have poured around 1 to 1.5 ml to on this specimen of the slide. So this we have to keep for about 5 minutes, and then we will discard the stain, the excess stain. We will discard the excess stain. So that we have to keep waiting for 5 minutes. And so our stain is over, and we will discard the stain, and just you can see it has taken up. The slide has taken up the stain. Now it is excess stain. So this we have to remove by bluing. This is the process is bluing, and this process is very important. So for this, what we are going to do is take around 1 ml or 2 ml of ammonia. This is our ammonia solution, our bluing agent. Just take some ammonia solution and pour it over the slide. You can see the fluorescence color has taken up, and this is our bluing process. So this will keep for about 30 seconds. We will just keep for 30 seconds, and we will discard the whole thing. So this is our slide is ready. So now we are going to give the counter stain. That is our rhodamine auramine confocal stain. So this will act as a counter stain. So some of the counter stain, what is this counter stain? Counter stain is it is the opposite stain. Some of the places which take up the positive stain, and some of the parts of this slide will take up the negative stain. So this is the counter stain or the negative stain. So that we will determine under the fluorescence microscopy. So this we will just take the soda stain, and our counter stain is almost ready. So this will be around 1 minute. We have to keep it for 1 minute, and our counter stain. This is our 1 minute counter stain, and we will discard this counter stain. So this we will again blue. Again we have to blue, and you can see this is again we have to blue. With the help of this ammonia solution, we have to blue, and see the stain is taking place. So this is also around for 30 seconds. This is for 30 seconds, and we will remove the whole thing. So now our staining process is complete. Our staining process is complete. So this is our staining process, and this we are going to visualize under this important. This is our fluorescence microscope, that EFL microscope. So this we will have to keep for, again we have to keep for drying, because this wet slide we cannot put in the microscope. As I have told you in my previous videos, that wet slides cannot be taken in the microscope. The microscopes will be damaged. So we have to clean the microscope, and we have to clean the slide, dry the slide, and then we will be able to put it under the microscope, and we will be able to see. So this is all about the fluorescence staining video, fluorescence staining, and the processing video, the fixation processing video. So this is all about our 9 tutorial. Thank you very much for watching this video.